So, welcome everyone. Today is the second session and the last one. So, today we have again 3 speakers and a 30-minute discussion after the different presentations. But first, I will give the floor to Marimete to introduce the speakers and the topics for today. Marimete, the floor is yours.
Thank you, Anna Maria, and welcome everybody to this joint OptFORESTS EUFORGEN webinar. And for those who didn't attend the webinar yesterday, I just want to make a small recap. So OptFORESTS is a European project. It's led by INRA in France and funded through the Horizon Europe program. And one of the activities in the project focuses on evolution-oriented management of declining forest and restoration efforts. And as part of this work, we decided to arrange this webinar to get a broader perspective on how to detect genetic decline in natural populations, combining theoretical perspectives with case studies from different groups of organisms. And just to shortly mention a little bit about yesterday, because I think it's relevant for the discussion afterwards. We had 3 speakers from the nature conservation community. And Alicia Mazzaretta-Janes represented the work done on assessing genetic indicator for global diversity framework. And we discussed the importance of having these indicators. Although they may not meet the gold standards, they are important to achieve policy and management impacts. Geir Bolstad talked about gene flow from farmed wild Atlantic salmon and how they monitor genetic introgression using specific genetic markers and how they are using this monitoring in management of locally adapted wild salmon population. And we also discussed the effect of gene flow beyond the wild salmon genetic integrity and if heavily one-dimensional gene flow can influence effective population size in salmon. Ophelia Ronch addressed whether assisted gene flow can rescue population at risk, and she emphasized that there is an optimal level of introduced genetic variation for evolutionary rescue, and that, for instance, late intervention may lead to the loss of local diversity. And in the discussion related to threshold for genetic decline, I think there was an agreement on setting this threshold is very difficult as they often reflect value-based decisions about the desired level of conservation. And when it comes to research gaps for identifying critical thresholds, what remain poorly understood is maybe how genetic diversity relates to ecological resilience and how genetic variation interacts with plasticity. So I think that's also interesting for forestries. We discussed how to genetically enrich declining populations, for example, populations strongly affected by drought, and Ophelia emphasized that it's important not to do enrichment blindly, but again, we don't know exactly what will be adaptive in the future. And when we enrich, we should be quite cautious, not losing local genetic diversity and homogenize genetic diversity. And also the timing of enrichment may be very important, and there may be a window when we should do this genetically enrichment. And today we will learn more about the practice related to genetic diversity in forestry and how forestry genetic community evaluates genetic change. Maybe one of the discussion topics. Is there any special about forestries? And what are the possibilities and challenges related to breeding and ex-situ conservation strategies? So the first speaker, as you see on the program, is Marjana Westergren. She's a forest scientist, a population geneticist, interested in gene conservation and use of forest reproductive material. And Marjana is working at Slovenian Forestry Institute, and you see the title of her talk in the slide. The second speaker is Arne Steffenrem. He's a forest geneticist specialized in quantitative genetics and breeding, working at the Norwegian Institute of Bioeconomy Research and the Norwegian Seed Center, the latter being responsible for the breeding of forest trees in Norway. And the third speaker is Jan Petter George, also a forest geneticist with interest in gene conservation, local adaptation, and forest monitoring. And he's a senior researcher at the Natural Resources Institute in Finland. And the discussion afterwards is going to be led by Erik Kjær from the University of Copenhagen. And Erik is also the leader of the work package Breeding with and for Genetic Diversity in OptFOREST. So that was a small introduction. And Anna-Maria, please.
Thank you very much. So without further ado, I would like to give the floor to Marjana.
Thank you, Anna-Maria. I would like to share my presentation. Like that. And can you see it?
Yes.
Yes. Okay. So thank you, Marimete, for this really nice introduction. Yes, I come from Slovenia, and Slovenia is maybe a little bit different from other European countries because our forests are mostly naturally regenerated. And I was trained as a forester in this forest, and I am interested in how the processes are working and how do we mimic this natural regeneration when we use forest reproductive material. So I will be talking a little bit about that today, and I will be talking in specific how evolutionary change happens in natural forest stands. You all know this, that under climate change and other threats, the tree populations can migrate. They can adjust through phenotypic plasticity, they can adapt or go extinct. And the question here is whether populations can adapt fast enough through evolutionary change. So you will see if I answer this partially, I will not answer it completely. But you will see through my presentation that this is a very complicated topic. So I will be talking about first the components of evolutionary change, and then I will delve into 3 case studies on European beech, on maritime pine, and on narrow-leaved ash. And I will be talking within these 3 case studies about effective fecundity as a fitness proxy. The second thing I will talk about is selection gradients and heritability. And then at the end, a little bit summing up about the perspectives and how to manage genetic diversity. First, about the 3 components of evolutionary change. So the first component is phenotypic variability. This is because natural selection acts on phenotypic variability in the field. Of course, then there is selection, and that we calculate here or term it as a selection gradient, which is a gradient of a regression of fitness on a trait value. And the way we estimated selection gradients was via parentage analysis using realized fecundity as a proxy for fitness. Now, a significant selection gradient can make a population evolve, but only if the trait in question is heritable, which brings us to the narrow sense heritability, the proportion of the trait variance that is due to additive genetic factors. And all 3 components are needed for a population to evolve. So let's go to the case studies. The first one is on beech. And actually, that is the only one that was already published. And I will only skim the results because it's a very complicated study. So if you will want more details, I welcome you to read the publication. So in this case study, we sampled 250 adult trees and then 200 seedlings. And these seedlings were sampled at different time points and at different ages. So they were 1 year old, and then they were from established regeneration 5 to 9 years old. And to calculate the effective fecundity and coancestry matrices, we used microsatellites. Then the phenotypes we have measured were related to growth, to phenology, and to reproduction, flowering and fructification. And we measure these phenotypes in multiple years. The second case study is on maritime pine, and here we were working in two contrasted environments. In Atlantic environment, which is less severely, which is a milder environment, and in Mediterranean environment where there is stronger pressure with drought. And the sampling design and the study design was very similar to the beach one, except we had more trees here, 500 adults, 250 seedlings, again from established regeneration. And here we used SNPs for molecular work. Now also here the phenotypes were related to growth and these were measured, but then we also had some innovative phenotypes that were calculated from calibration with the near-infrared spectrum. These were specific leaf area, leaf minimum conductance, and loss of hydraulic conductivity. And the results of this study are under preparation, and I hope we will publish it soon. The third case study is on narrow-leafed ash, and this study was slightly different in design. It included 5 different sampling plots, sampling sites along the Danube River and its tributaries, and the number of trees sampled here were smaller, but again we focused on established regeneration only, not on 1-year-old seedlings. Here we also use SNPs, and the only a phenotype that was scored was disease severity, and I'm talking here about the ash dieback. Also, this study, the manuscript is under preparation. Okay, so let's look at what we found out about effective fecundity, which was our fitness proxy. In beech, we have seen that the effective fecundity was highly skewed, meaning that we had a few trees that had a lot of offspring, and then slightly less trees with fewer offsprings, but then there was a lot of trees that didn't have any offspring at all. And what you can see here also that there are slight differences between the female and the male function, and that there are differences at when we measure this effective fecundity, when we measured it on established regeneration and in 1-year-old seedlings. So fitness of trees all change depending on the life stage. In the second study, we have seen that fitness also changes across the environment. I will not talk more about this, but you can see it on the left graph. We use the mixed effects mating model to primarily to calculate the effective fecundity. Okay, forward to the selection gradients and heritability. For beech, we were thinking long and hard what we would expect to happen in terms of selection in beech in the environment we had the plot. And at the end, we hypothesized that most selection will be directional. This would be for tree site, for the seed amount, for bud burst, and for leaf senescence based on what we know about the species and the environment. And then we thought that the flowering would be under stabilizing selection around an optimal point in time. And then what we have seen is slightly different to our expectations. And you can see that here I depict selection gradients for seed, for 1-year-old seedlings in blue, and then for established regeneration, depending on when the traits were measured, either in 2018, which was a mast year, and 2019, which wasn't a mast year. So those are the two green colors. You will also see that some of the colors are lighter in shade, and that is because the selection gradients were different for the male and female functions. And where you have 2, it means that the selection gradient was quadratic, so stabilizing selection, and +2, it means that it was linear, but also with a quadratic component. So what you can see here, that not all traits there were not significant selection gradients for all traits in all life stages. And also what you can see is that they sometimes differed from our expectations. For example, not for the tree size, but they differed, for example, for the flowering or for the bud burst, and also for the seed amount. So, and when I started talking, I said that the traits also have to be the traits have to the traits also be heritable if we can expect evolutionary change. So, and then it turned out that most of the traits they had significant selection gradients, but they were not heritable. So we can only expect some evolutionary change for certain traits, and these were for growth and for bud burst in spring. If we now go to Pinus pinaster, where we looked in 2 populations in contrasting environments, we can see that here again, like in beech, we had selection gradients for growth traits, and the only selection gradient that we had for functional traits was the one for 50% loss of hydraulic conductivity, P50, which is very much related to drought. And we see that the selection gradient here is showing a stabilizing selection around an optimal point where trees can resist drought. And again, if we are looking which traits were heritable so that we can expect evolutionary change, we can see that the DBH was heritable like in beech, but then also this P50, which is a very important trait, was heritable as well as SLA. But we can only expect evolutionary change then for growth and for P50, because for the other 2 traits that were heritable, we didn't have a significant selection gradient. If we now go to the narrow-leaved ash, here what we have seen is that genetic diversity was maintained after selection. Now, maybe here I have to say as well that the ash dieback came into the area in 2007, and that we were doing the sampling in 2019. So there has not been really a lot of time that has passed between the onset of the disease and the time of sampling. And at the time of sampling, the seedlings were also 5 to 8 years old approximately. So, but in that time, we didn't see any change in genetic diversity measured as expected heterozygosity or in percentage of polymorphic loci. Then what we have seen is that there is strong genetic control for resistance to ash dieback. The resistance was highly heritable. And we found 29 SNPs that explained 45% of variance, so the difference in disease severity in the trees. And here we used lasso regression for this work. So then these 29 SNPs, we call them the positive effect alleles, and the number of these SNPs that each tree harbored was negatively correlated with disease severity. But what we also saw is that the number of these alleles was similar in both adult and the seedling cohorts. So there was no apparent change between the cohorts. So despite the despite the strong heritability of the trait. And maybe this has to do precisely with the fact that disease was not present in the area for a very long time. So maybe the phenotypic variance was not really high yet. Trying to reflect on these studies and summing up what we can learn from them. The first thing I can say is that standing genetic and phenotypic variation are crucial for evolution. And also something that I found really important is that relative fitness was difficult to measure, and it also depended a lot on when it was measured, whether it was measured based on seed, on 1-year-old seedlings, or on established regeneration. Then also what we have seen is that we have genotype by age interactions, and that natural selection can act in opposite directions at different ontogenetic stages. Now, there was also high selection probably between seed and 1-year-old seedling and the sapling stages, because selection gradients turned from not significant to significant later in life, or vice versa. And then also, of course, we had genotype by environment interactions, because and we have seen that selection pressures can vary from year to year following the environmental variation, and of course the environment also on larger distances modulated the selection. So looking at these studies, what I would like to point is that we should nurture diversity in forest and nurture the variation that we see. And maybe it would be good to extend natural regeneration over multiple masting events, because things are changing and happening, and they go in different directions depending on the years and what is available. So I think we should extend the base, the genetic base on which the evolution can act upon. And what I also think would be wise is not to judge until the initial selection phases have taken its course. And then I was thinking how to replicate this selection in planted seedlings. I don't have any answers here, but maybe planting them from multiple masting events or multiple seedlots. But we have also heard from Mari Mete today that there is a window of opportunity when to do that, and especially if we mix seed from provenances that are far away, not to swamp the local gene pool. Also, one thing that I thought about is increase the planting densities, because then we give the opportunity for natural selection to act upon these seedlings. Yeah, I don't have the answers here, but on this note, I would just like to thank all the people who have worked on these 3 case studies. There was a big team of people on each of the studies, and also the projects that funded these studies. The beech study was funded by the LiveGenMon project, the Pinus pinaster one by Forgenius, and the ash one by the Interreg Refocus project. And with that, I would like to thank you for your attention.
Thank you very much, Marjana, for your presentation and your interesting studies. We have now 5 minutes for questions. In case we want to have clarifications. Darius?
Yes, it was an interesting study. Just a short question was that have you been thinking to find or to check for any associations between your molecular data and your phenotypic data, such like phenology, for instance, to have an indicator of genetic diversity other than DNA marker? And I go for genetic monitoring, you know, as you did it also in LiveGenMon project, and we are sort of looking for another indicator than DNA, sort of indirect indicator of genetic diversity. to be used for genetic monitoring?
We have tried, but in LiveGenMon we have tried, but we used microsatellites and we couldn't kind of connect this to phenotypes. We haven't seen anything like directly that different diversity would be associated to different phenotypes. No, we haven't really seen that apart from what I have presented in this study.
Okay, thank you.
Thank you. Mari-Mette.
Thank you, Marjana, for a nice talk. I was just wondering the heritability estimates for ash.
Yes.
Is that based on trials or in
No,
situ
it's it's based on kinship. We calculated kinship from the regeneration and yeah, we calculated kinship. So
missing because we are doing a little bit of the same in OptFORESTS, both Lenenings in Denmark and here in Norway as well. It will be exciting to see the results. Yeah.
Okay.
Nice,
great.
promising. Yeah.
Yeah.
Thank you. Any other questions? I don't see anything in the chat. Erik.
Yeah, thank you for a very nice presentation. So just the point, it seems for me that when it comes to the ash dieback, your conclusion would be that there's a high heritability in the site, and you say you don't see the signs of the response yet, but probably because it takes time.
Yeah.
But while for the beach, it seems was the conclusion that their natural selection is working much weaker, or is that fair to say that? So it really for some traits, but for many traits, actually, it seems like selection is working quite slow.
Exactly, and then when I didn't go really into details, but we calculated the responses on the traits that were, you know, that were evolvable, and the response is actually like, you know, in a generation, a centimeter or even less in terms of growth. So very, very small responses to selection responses. So
do you think that is? So do you think that is because that there actually is basically you are dealing with stabilizing selection because at the point of departure, populations are actually adapted to the best, you know, or is it because you think that is just so slow?
I mean, I don't know. In the beach study where we have measured this, it's a pretty optimal site for beach. So I would say that our population was quite well adapted to the area, and that's why we see slow response. And in ash, for example, I think the heritability was really high, and I think that we will, if we go again, you know, in a few years to the same area and we check the trees that have survived, and I think we would be able to see something much better. I think the study has been done a little bit too early. So there were some differences in not significant, but there was a trend with the populations that encountered the ash dieback slightly earlier than the others. So yeah.
Okay, so thank you. Yeah, so it makes sense that there has to be some kind of selection going on, otherwise you wouldn't expect to see a response. Okay, thank you.
Yeah.
Thank you, Francois.
Okay, thank you. My question is, do we, because the selection that you measure is based on the reproductive success, but all the reproductive trees are already the result of selection. So you miss all the selection that happened before, and maybe at this stage there is no much room left for this residual selection, but maybe high selection has already occurred in the previous stages before reaching the reproduction stage. So we probably underestimate with this approach the selection intensity, no?
Yeah, I think it's looking at the site long term. Yeah, I'm sure that we are underestimating, but when we are just looking at what is happening currently, yeah, then this is what it is, and it is for sure affected, but by what has happened before, I would say. Yeah. So I would expect much higher in more stressful environments, or if something new comes like the ash dieback, which we haven't encountered before. So I would expect stronger responses in cases like that if the traits were heritable. Yes.
Thank you.
Thank you very much, and thank you very much, Marjana. Let's move on now to our next speaker. So Arne, who is going to talk to us about breeding. Arne, the floor is yours.
Thank you. Well, thanks a lot for inviting me to talk about the breeding programs in Norway that we might use as an example for the further discussion. This topic that I've been given is quite closely related to population genetics, and I'm not a population geneticist. I'm basically a forester with a PhD in quantitative genetics and tree breeding. So my interpretation of the population genetics that I present today is from the base of being a breeder that is responsible for implementing present knowledge into the breeding programs. Yes, this is actually my view from my office in Steinkjer here in mid-Norway. So besides from the windows being a bit dirty, that we have, at least last week, we had beautiful autumn colors in this boreal forest with lakes, both late succession conifers, clear cuts, early succession birch forest, managed forest, and unmanaged forests. This is a typical example of the forest structure in the more productive parts of Norway, mid-Norway particularly. Yes, in primary production, the temperatures and temperature sum is fundamental for the production potential, and the expected temperatures is going to increase. And I'm going to focus mostly on the most optimistic emission scenarios, maintaining around the temperature increase of around 2 degrees on the long term. But we all know that that's very ambitious and maybe not possible to achieve with the politics going, the politics are going today. But even with this optimistic scenario, we in Norway can expect an increase of the temperatures and temperature sum during the growing season of around 400 degrees, which is a lot when the average today is, or at least in the past, has been around 11,000 to 1,100. In many regions, that this will cause an increase in productivity, but in other regions it might cause more challenges than benefits. So this is particularly connected to our favorite conifer spruce, which is quite vulnerable to drought and the following bark beetle attacks when the spruce stands reach the mature age. So within the time frame that we are developing seed and bred material for now, we have to consider our insurance policy already now, which is both genetic variation and species variation. And that's why the new breeding strategy that we wrote last year in Norway Also consider more tree species than we have done earlier. The main goals of the area of the strategy is to produce area efficient forest reproductive material that produce high volumes of high quality timber, but it should also sustain the genetic variation on long term, and should provide forest with reproductive material that is robust to climate change. And we should have a nationwide supply of high quality seed. And as I said, we have this strategy taken in over us that we actually need to have seed supply and possibly also breeding for more species, as Norway spruce might become less important in the southern parts of Norway, while other species like, for instance, oak might be more important. So the new strategy contains also breeding plans for Scotch pine, birch and oak, which is new since we have had a great focus on Norway spruce until now. And this is it's particularly important for us to be able to supply forestry with seed, so forestry can actually perform site adapted species decisions and species compositions. A problem in Norway has been that we have planted spruce almost on all kinds of land, very often because the moose population has been so high that you cannot plant anything else. It's going to be eaten, while spruce is not a favorite meal for the moose. But the management of the moose is more strict now, and there are less moose. We also have some wolves in the eastern part of Norway, which also reduced the population of wolves and of moose. So the pine regeneration starts to develop nicely, which is new, which is quite new to us in many regions. So now it's actually feasible to plant pine instead of spruce, and that's particularly important on forest types where spruce isn't that adapted. Here in this picture you can see spruce planted on a pine site, and the spruce doesn't develop very nicely. In this presentation I'm going to talk about mainly spruce breeding, because that's the breeding program we have had the most experience with, and that's where we also are mostly concerned with genetic diversity and also adaptation. The core strategy for spruce breeding is based on the principles described by Øye Danell for the breeding conifer program in Sweden, and these are main strategies that are followed for I think all countries in the Nordic region in conifer breeding. The breeding population is quite large. It's around 1,000 individuals, and it's divided into subpopulations of 50 individuals that should cover the adaptation clients that we have in the country. So this is the figure from the Swedish breeding strategy. We show that the subpopulation, they cover the span in variation in temperatures, growing season, actually it's the growing season days, and then the photo period. The reasoning behind these subpopulations of 50 individuals is that with random mating within this group of individuals, we will maintain nearly all additive genetic variation from generation to generation. The breeders, they're not doing random mating since they are selecting for growth, but the breeders, they select only for a few traits. And since the rest of the traits are mostly uncorrelated with growth, we can probably assume that additive genetic variation isn't changed much more than what we see in this figure here. So with a population size of around 50, you will maintain nearly 100% of the additive variation in each generation, from generation to generation. We have adopted this strategy also in Norway, but we have been more concerned about the geographic structure because we also have the coast and the higher elevations in Norway. So we have subpopulations for the lowland, the mid elevations, and the high elevations. And then we are selecting new subpopulations from Eastern European provenances planted in Norway, which is going to be future breeding populations that are going to be utilized in a more futuristic environment. The reasoning behind this subpopulation structure is kind of it's a little bit vague. But what we know about Norway spruce in Norway is that it's a very recent colonization. There are high genetic variation within populations, quite little genetic variation among populations. There is strong gene flow. And many places we have only 2 to 3 generation spruce after the first immigration. So in this map here you can see the immigration, the time that it has been estimated that spruce has been present in the different parts of Scandinavia, and it's very young in Norway. And the immigration routes have probably been around north, around the Baltic Sea, and then from south over the Baltic countries, the Östersjön, and then into southern Sweden, southern Norway. These two routes might have caused 2 different adaptation developed, and that's also why we have been a little bit concerned with the structure in Norway, even though we haven't found much structure in earlier genomic analysis. We have kept these subpopulation quite geographically separated. But now we have done some new structure analysis based on the SNP chip data. We have a lot of these now from the breeding program, and since nearly all clusters and selections are being genotyped, and we can see that we have some grouping. They have the introduced material from Central and Eastern Europe, of course, but we also have some kind of a cline within the natural range plus trees. And based on this, we can see that mid-Norway might have a different, a little bit different. Yeah, the differences is not big, but it stands out as one, as a separate group than the southern Norwegian material in this analysis. But the differences are not big. But this might be, the background for this might be the different immigration routes mainly. So these populations I showed you on the map. If you look at the climate and photoperiod for them as yeah, you can see on the x-axis here you have in the figure to the left we have the average temperature sum, day degrees above 5 degrees Celsius for the 22 breeding populations that we have in Norway, the 22 subpopulations. And then the mean day length at the 1st of July, approximately. That's around when growth cessation and occurs. So you can see that there is a, I've added some variation to these dots to illustrate the within population variation that we expect to have. If we look at the figure to the right here, then we can see the comparison of the climate in the period 1980 to 1990 and 2010 to 2020 for these subpopulations. And we can already see now that there has been a shift from the 1980 to the climate that we have presently. It's become around 100 degrees warmer already. But if you look at the climate until 2050, 2060, following this RCP 2.6, the 2 degrees increase emission scenario, then we can see that the subpopulations here will probably experience 400 to 500 degrees higher temperature sums than that they were selected for in the beginning. So it will be a quite different climate, and then it's the growing season that will increase. The length of the growing season will increase. The photo period of this population will not change, of course. So that is why we also decided to select breeding material from Eastern European provenances that's already planted in Norway. And in this picture you can see a local provenance planted in this square to the left here. It was originally planted 4 by 4 trees in this plot. So it was 16 trees in this square. And then to the right here we have a Romanian provenance from the Carpathian Mountain, 850 meters above sea level. 16 trees in the plot here as well. And over 50 years, these Romanian provenance has completely outcompeted the local Norwegian provenance. So it's very evident that this more southern provenance is utilizing the longer growing season is performing a lot better in the climate that we have now. So back to the core strategy. Within the subpopulations of approximately 50 individuals, we select, we test and select the breeding material. The plus trees originally selected, they are tested, and then we select which is going to be the parents for the future generations. The testing is going on by mating, trials, measurements, and selection. And it's going for cycles or over generations. And the selections that we do is for traits with quite low heritability, like growth, stem straightness, and double leaders, ramicorns, spikenknots, etc. And then the best part of the breeding material is selected for seed orchards, and so the improved materials is propagated in seed orchards. We expect the breeding cycle to produce genetic gains of the plus tree seed orchards. We expect to produce genetic gain of 10 to 15% increased volume. But with the tested plus trees after the first cycle, we expect the gain to be around 15 to 25%. And then when we have finalized the second cycle, which will be in 20 to 20 years from now, we will establish seed orchard with a higher genetic gain. So this is a long time endeavor. The progeny testing is going on in multiple field trials. So we can test the different materials over a range of climates. And I can see that already now that looking at the climatic scenarios, we have been to careful about testing the material locally. We should have tested it over a bigger span of climates from the beginning, but they didn't consider that in the 80s and 90s. Then we do the phenotyping and selection after the age of around 15. Then we start selecting, but we also utilize the same trials for many more years. So if we have old trials that have experienced more time and more climate, we do selections in them as well. So you can say that the parents, they were superior in the 1800s, and the progenies are superior from the 1980s. So these families, they have performed well in both the new and the old climate. We have been testing whether we maintain how much genetic diversity we deliver from the first generation seed orchards. We haven't had many seed orchards established from the second cycle of breeding, first or second cycle of breeding. So our tests of the outcome in terms of genetic diversity has been for these first generation seed orchards, which is mainly plus trees. We tested 2 seed orchards with microsatellites in this project called SustBreed. One seed orchard with 60 clones, the other one with 25 clones. We studied parental contribution, pollen contamination, effective population size, allelic richness, and genetic differentiation. We found that there was a pollen contamination around 20%. The parental contribution were very uneven, and the effective population size were 60 to 70% of this consensus size. There wasn't much genetic differentiation between the seed orchard progenies and progenies from the natural stands. But the allelic richness of this seed orchard with 25 clones were a little bit lower than the allelic richness of the progenies from natural stands for the representative regions. However, this seed orchard with 60 clones had approximately the same level of allelic richness as the natural progenies. So when breeding has developed, we have also got new tools for the breeders. And we are actually using these new genomic tools, these SNP chips that has been developed for Norway spruce and Scotch pine, almost in our daily work now. We genotype parents and progenies in the breeding population. And we have a very nice pipeline of work with collection, storage, DNA extraction, genotyping, and then analysis and storage of all data in a database where we connect all phenotypic and genomic data. So we have a nice workflow for that. And then we can actually use this in breeding. So we can improve the breeding values when we have the opportunity to use genomic and pedigree data together. We can do optimized selection from progeny trials, particularly in progeny trials when we have open pollinated progenies from seed orchards, or we have pollen mix families from these pollen mix matings that they did a lot in the 80s. They had a mix of 16 parents in a pollen mix, and then they made control crosses with that. And when we have clonal archives with more or less relatedness, we can also do the optimized selection and optimized mating design based on the G matrix. So we have, when we have the breeding values and the G matrix for all individuals that are candidates for going into the crossings, we can run an optimization program. We use something called simple mating to select which crosses to do. And that's very convenient because in the mating season, everything is in a hurry. So we don't have time to do much manual data analysis. We have to do it quick. So to summarize, how to improve adaptation and avoid genetic decline. Guess that was the question I got for my presentation. We must distinguish between the breeding population and the seed orchards. That's very important. The breeding population is where you maintain long-term the genetic resource for breeding. This should be large. We have around 1,100 individuals in our spruce breeding population. That is going to be parents for the next generation. We should allow assisted gene flow, and I'll leave it in the air how to do that. We should avoid unintentional breeding. We should utilize genomic tools to increase flexibility, accuracy, and efficiency, and also control this in breeding. We should test in the relevant future environments. And what is very important is that the breeding organizations should be long-term, have a long-term responsibility, and it's important to keep the competence high there. And the seed orchards should contain sufficient number of parents. It should be designed so we promote large variation in crosses. We should balance gain and pollen contamination. So if you have little pollen contamination, you might need more parents in the seed orchard. You should differentiate the parents between seed orchards for the same region, so you get a mix of parents over the landscape. You should also facilitate assisted migration. You should have data so you can use the seed orchard material further north or higher up if it's necessary. And maybe we also should consider pollinate our seed orchard with a mix of introduced pollen from areas that more represent the future environment. This was a paper that we published this year actually with David Glilil and Milan Lisburek from Czechia. So with that, I'm gonna say thank you for letting me have this presentation.
Thank you very much for your presentation. We will have time for 2 questions, and I see 2 hands. So Darius.
Yes, it was interesting, and of course, there's a lot of things to ask. It's a pity time is short, but the major things I wanted to ask you: have you decided to test cuttings like they do in Sweden and Finland? Because it's a really effort, you know, in your breeding population for Norway spruce. You know, you make the crosses, get the progeny, and then the question is that we were trying to think for a long time: shall we test the progeny itself and select by phenotype, or we clone each of those candidates and then establish those cutting trials and clone by cuttings? And that means huge effort. I mean, then increases your testing population increases for many times. So have you decided to go for cuttings in Norway spruce, or you test the progeny after the crosses itself? I don't know, I have more questions, so I can go with just one question more that would be very interesting, but if you could respond to that, then I could have another one.
Yes, we do the cuttings, we do. So these multiple tests, they are also, we are also utilizing the cuttings to test the same genotypes over different environments. But
Okay,
not in all,
okay.
but not in all cases. We combine the 2 methods.
Okay, then there was another about these SMP markers which you are using for your assisted selection. Can you tell what is the set? Is it that your own that you made it, or is it like those standard ones that we can get from
It's
companies?
the standard ones.
And which one is that? Is it this 50 key or?
Yeah, it's the
Okay.
50K. We use the Finnish one for Scotch pine and the Swedish SLU one for Norway spruce.
Oh, interesting. And of course, you would be interesting with assisted migration. And I saw your breeding populations that you have two from Eastern Europe. Interesting. Do you have any from Baltic states, and how you think to integrate those foreign breeding populations into your mating population? For instance, you mentioned about this supplementary pollination, but maybe some clones into seed orchards, or do you have a strategy how to integrate those eastern breeding populations into your seed orchards?
Yes, we do have Baltic materials. We as well. I think the most of the material from Eastern Europe is from the Baltic and Belarusia. And we use progenies from these provenances in seed orchards already, and hoping that they will be sufficiently synchronized with the Norwegian material when they're going to flower.
Yeah, that could be the problem that we noted, that those, they flower in different times when you move them for, I think, 2 degrees of latitudes, and even more, than they will differ in phenology. So that was my sort of concern. We are thinking also about our breeding populations in a similar way. We take them from Poland, and we have this question of not really mating. So yeah. Okay, so give for the other people. Sorry for long.
Thank you. Thank you. Erik, a very quick question because we have to move to the next presentation.
Thank you very much for a very interesting presentation, Arne. A very short question. You showed that already now you have, you can see that the climate already now actually moving outside your historical climate in some places, so you cannot really find any local seed sources that would actually fit. But then also, you, of course, within this movement of climate, so to speak, there could be now cases where you have things which is adapted to a warmer climate that you can then use in Norway in the present climate. So are you doing that when you said about you can use your seed orchard for seasonal migration? Are you already now starting to move to use seed sources in different places to as a response to the present climate change?
Not really, because our seed orchards are dimensioned and the size of them is fitted into the area of use where we expected to use them. So if we start to transfer them too much north, we will run out of seed in the south. So we haven't really started migrating the seed from the seed orchards yet. We haven't done that. But that's definitely something that we need to make a strategy for. Maybe even moving the breeding populations northwards.
Okay, thank you.
Thank you very much. Thank you very much, Arne, for the presentation and for the questions. Let's move on now to our last speaker for today, who is going to talk about dynamic and static situ conservation. Jan-Peter, the floor is yours.
Yes, thank you. So now you can see it in presentation mode, or?
Yes.
Yes. Okay. So thank you very much for inviting me and giving me the opportunity to speak here. You will see during the course of the presentation that my presentation will be different in many aspects from the previous ones, but I hope nevertheless interesting. So the title of my talk is ex-situ strategies for effective gene conservation and European forests, equivalency versus complementarity. So as a brief overview of what I will be presenting here, I will talk a little bit about common and new definitions of ex-situ conservation, and then present a few case studies to make the entirety of ex-situ approaches a little bit more tangible. And after that, I will discuss indicators that can be used to monitor conservation efforts at national and international levels. So yesterday we heard a lot about indicators of genetic decline, and today we will look at the other side of the coin, namely how well populations and species are conserved against genetic decline. And I will conclude with a very striking slide that will show that there's still a lot to be done in the area of ex-situ conservation, and also show how in situ and in-situ and ex-situ conservation must work together in the future to conserve forest genetic resources most efficiently. But before we dive into the technical details, I would like to mention that the results I'm presenting here are the outcome of a young and very dynamic working group that has been racking its brains over this topic for the past 2 years, and the text version of this 2-year brain racking will soon be available in the form of an official report. First of all, why is there a need for clarity when it comes to ex-situ? As part of the working group, we sent out a survey to all EUFORGEN contacts, and here are some of the results. And as we can see on these 2 maps, there are still major differences when it comes to seed banks and clone archives, for example. And this is even more evident when it comes to how and where material is conserved, as you can see here on this bar plot. And this makes it clear that we need to ensure uniform data standards and also define best practices if we want to harmonize and monitor all these efforts at a European level. But before we even begin to talk about ex-situ conservation, we need to clarify how it differs from in-situ conservation and how we can differentiate within ex-situ conservation. In Europe, we generally differentiate along 2 dimensions, which relate to location of conservation and the type of conservation. For in-situ conservation, there's only the dynamic option, as it involves generational change on site, in which as many trees as possible participate and can enable adaptation to gradual environmental gradients. For ex-situ conservation, however, it becomes more complicated. So we distinguish between static ex-situ, which you can see here on the right, which refers to the permanent conservation of germplasm, either in seed banks or cryoarchives, or the long-term storage of pollen as haploid genetic material. But then we have something classified as dynamic ex-situ, which includes, for example, conservation seed orchards, where we artificially bring together many individuals that would be less likely to exchange genetic material in their natural habitats. And another curiosity is the transfer of genotypes between different environmental zones within their natural range in order to promote adaptation to new climatic conditions, for example. And this includes any form of assisted migration, assisted gene flow, and also assisted colonization. Why should we even distinguish between in-situ, dynamic ex-situ, and static ex-situ? Well, the most obvious reason is that the different systems do different things. While dynamic in-situ and dynamic ex-situ are very good at maintaining gene flow processes and producing offspring with high diversity, static ex-situ measures are more suitable for preserving genetic resources in the long term and over large areas from bottlenecks such as pandemics, for instance. A good example is the seed bank operated by Norwegian in Svalbard, where also forest genetic resources are stored for rather apocalyptic scenarios that we all hope will never occur. At some point in the future, we will have to consider how these different systems should be evaluated and organized. They can be organized either as equivalent systems, as you can see here on the left, as partially complementary, or as strictly hierarchical, whereby dynamic in-situ continues to be seen as some kind of golden standard, and in certain cases is backed up by dynamic and static ex-situ. But I can tell you that there are as many different views on this as countries in the European Union, and therefore much broader discussion is needed before a clear answer is found. At this point, however, it is worth looking at a few case studies to get an impression of how diverse ex-situ conservation can be. The first example takes us to Finland. White elm is one of the rarest and most endangered tree species in Finland and is strictly protected by law. And we conserve genetic resources of white elm in a combination of living collections, which is dynamic ex-situ, in which genotypes from the entire distribution are brought together, and static collections in which we store the same genotypes in cryotanks for the long term, hundreds of years if necessary, and then we thaw them up and clonally replicate them as needed. And this has been proven a very efficient conservation strategy as it combines dynamic and static approaches for species which would rarely thrive in gene reserve forests, for example. Second example goes beyond Europe and deals with an extremely rare tree species from South America, the Patagonian cypress, which is highly endangered due to human overuse and is therefore listed in the Washington Convention on International Trade in Endangered Species. And this tree species had been conserved in botanical gardens in the British Isles for a long time, but it turned out that all individuals conserved ex situ were descendant from a single female clone. And in order to expand the genetic base of these collections, it was decided to sample additional material from across the entire distribution area and conserve it in a total of 60 locations in Europe. And this is a good example of dynamic ex situ conservation with a strong focus on nationwide networks of collections, which are always well protected in the event of the loss of individual clones. The last example takes us to France and deals with wild cherry, Prunus avium, which has high economic value in France due to its wood. And in France, so-called synthetic populations, which are created in a biogeographically stratified manner for different ecozones, enable the wild cherry tree to produce offspring with high genetic diversity and ensure that natural regeneration in the stand is guaranteed through management of these populations. And this is one of the key differences to clone and family archives, for example, which can potentially produce forest reproductive material, but it is rarely clear what this forest reproductive material is used. So in this example from France, it is ensured that the material is used on site. Okay, and this brings us to the last part, which is about indicators. So yesterday we have heard a little bit about indicators which are able to monitor genetic decline. But of course, we also need indicators which can effectively monitor conservation success so that we know how well populations and species are protected against genetic decline. And these indicators also support harmonized and coordinated national conservation actions towards a pan-European strategy. Oops. Yes. So there are already indicators in place, and probably some of you have already heard about them. So usually when it's about dynamic in situ, then we monitor species diversity, which is the number of species listed as target species in genetic conservation units divided by the number of species occurring within the country. We have ecozone diversity index, which is the number of ecozones represented in the National Conservation Network, again, divided by the number of ecozones occurring within the country. And we have something which is called an insurance index, which is the number of ecozones represented in the conservation network with a minimum number of 2 units divided by the number of ecozones occurring within the country. So all these indices are standardized to 1, and so they vary only between 0 and 1. And they are able to effectively monitor the conservation status of species in Europe. So here's an example of this indicator, indicator 4.6 for European ash, Fraxinus excelsior. So the yellow dots on the maps are dynamic in situ conservation units. And you see that this triangle here shows that how many countries are involved in conserving ash, how many ecozones are conserved, and then how many ecozones we have more than or equal or more than 2 units. Well, what is the problem with this indicator? The problem is that, well, as we all know, ash dieback is currently highly impacted by ash dieback. So in many of these units, we can assume that there is a very severe decline in population size. But of course, this indicator does not tell us anything about it, simply because it cannot be measured. So what we saw in the working group was a need that we also try to translate these indicator 4.6 to monitor dynamic and static ex situ. So what we did is we gathered data from 6 countries and for 6 species. And then we simply did the math and tried to calculate indicators similarly to indicator 4.6 to see how complex it would be to monitor conservation success. And the simple answer is that, well, we were able to calculate the indicators. So we can potentially monitor dynamic ex situ and static ex situ at country level and also at species level. So here are 2 examples. One is for oak and one is for Scots pine. So what we saw for this preliminary data set for 6 countries was that obviously the only conservation effort for oak is dynamic ex situ, which makes sense because oak is a recalcitrant species. You cannot store seed for a very long time, whereas for Scots pine, it is rather a mixture of static ex situ by, for instance, seed banking and dynamic ex situ, for instance, in living collections. And we can also do this at country level. Here's an example for Norway, where we can see, okay, what is the effort in conserving species, how many ecozones are conserved, and what is the insurance. And we can also break it down for separately for dynamic ex situ and static ex situ. One of the challenges, or not challenges, but one of the questions is how do we define ecozone diversity and insurance for ex situ measures? Because for dynamic in situ, it is easy because the location of the site and the location of the material is identical. But for ex situ, it can be different because we have, in many cases, we have many different origins put together in one unit. And then it's the question, are we interested in how many geographical origins we have conserved or how many units we have established? Potentially, with the minimum requirements that we defined, it would be possible to calculate both, and we can also monitor both, as we can see here. So we can, for instance, calculate the average number of origins per species and also the total number of GCUs, and then we can monitor how these numbers develop over time, for instance. Again, the question is which indicator would make most sense, but this is something which the let's say European community on conservation of forest genetic resources still needs to resolve. We just showed up to now that it is possible to do it in an equivalent way to indicator 4.6. Okay, and then this is already my last slide, which is about knowledge gaps. So one thing that, of course, was beyond the report was the question, how do we see these 3 different approaches anyhow? So at the moment, we see them largely as separate approaches, which could be valued in a separate way. But I think the big question is for the future, if we should rather try to see them like really in a complementary way. So this is just an example of how these 3 systems could work together. Dynamic ex situ could be used also to fill up static ex situ repositories. And of course, static ex situ repositories can also be used for restoration of dynamic in situ units under decline, for instance. And they can also be used for translocating material between environmental zones to match future climates, for instance. So this is just one very simple example, and I guess we have as many possibilities as this little cube shows here. So this is something which we were not able to resolve until now. What kind of combinations of dynamic ex situ, static ex situ, and dynamic in situ will help us to conserve genetic resources most efficiently. Because having three systems as three separate systems could also cost a lot of money. So this is something for which a lot more research needs to be done in the future to figure out how they really complement each other. So as I told you, this presentation was a little bit different compared to the other ones. I hope it was nevertheless interesting. And yes, let me know if you have any questions. And again, a big thanks also to the working group for this 2-year ambitious work, which will hopefully lead soon to the publishing of the report. Thank you very much.
So, any questions for Jan Peter? Erik.
So thank you for a very nice presentation. So I actually have questions related to this dynamic ex-situ conservation that I think is a very interesting concept that you present. So you said that you have your conservation seed orchard. You consider them as dynamic ex-situ conservation. So my question, where will selection take place, so to speak? I mean, how would they be dynamic in terms of continued adaptation?
Yes, well, this I can tell you was also a big discussion within the working group. Well, the thing is, when we define dynamic ex situ, it is rather about purpose and potential. It is not about what is actually happening, because we also define in the same way dynamic in situ, although we have basically no idea where selection is taking place in the unit, right? But there's potential for selection. Because, for instance, you could use the seed from conservation seed orchards for restoration projects, and then selection would essentially take place there. If I had shown you the example from France about the synthetic population, this is a special case because the offspring is directly exposed to selection because it's growing on site. Whereas for other conservation seed orchards, the big question is, okay, where will you use the seed in the future? So the question is totally justified. But as I said, that's rather about potential. So the definition is rather about potential, not of what is actually happening with the material.
Super, thank you. And just taking this one step further, in OptFOREST, we work with this idea about breeding with and for diversity for the so-called minor species, and I say minor species citation because I don't like the word at all. But this idea of species that are used very little today, and you showed the example with the ulmus from Finland. And so my question would be, to what extent do you see development of seed sources and seed procurement programs for these species as actually a way of dynamic ex-situ conservation?
That is a very good question, and I can only give you a national answer from Finland. I have no idea how it is handled in other countries, but we have already started to design our new living collections and conservation orchards in a way that we can, so that clones or family members are assembled in a way that we can be sure that we will get out, most diversity out of this when we produce seed. And then, for instance, use the seed in, let's say, funding schemes and funding programs, which are about establishing new stands with higher species diversity, for instance. So we had a program, a project here a few years ago, which was exactly about this thing, thinking about how to redesign conservation seed orchards and living collections in a way that they can be used as seed orchards for providing forest reproductive material. But there are some obstacles in this, so it needs a complete rethinking, because at the moment when we assemble, for instance, ramets in the conservation seed orchards, we do it usually randomly in the best of all cases. But as you know, for instance, there are new algorithms like minimum inbreeding or optimum neighborhood design, which you could apply to conservation seed orchards in the same way as to a breeding orchard, for instance. And we have just established last year a new Tilia cordata conservation seed orchard in Finland, where we applied minimum inbreeding design. So we have at least an argument in the future to say, okay, when we use the seed from this conservation seed orchard, it is somehow ensured that it provides the maximum genetic diversity that is theoretical possible. I don't know if this answered your question.
Yeah,
but
yeah, super.
Yeah,
thank you.
Yeah.
Thank you. Yeah.
Thank you very much. And Darius?
Yes, I was thinking to ask you, have you been, maybe it's not really in line what you said, but the idea was to ask, have you been thinking about the sort of measures to enrich genetic diversity in your forests and also maybe in your ex-situ seed orchards? You mentioned about this mating design and so on, which is quite possible, but also for mating designs, the things are complicated when you have an equal representation of rames from each clone, then it complicates matters a bit, but that's another question. The major question would be, have you been thinking in Finland in general, what measures you would be using to enrich genetic diversity in your forests, for instance?
Well, I cannot say very much about this because this is essentially the section of forest management where I'm not really involved in, I have to say. I see myself, and I think also most of the community, as that we provide somehow the raw material for this and the good basis, and how this material is employed and what kind of silvicultural practices and schemes. I think this is really part of the silviculturalist. I think we must work together with them in the future to figure this out. But as I said, I see myself more in a position to provide a good basis. But then the rest is really about silviculture, I think. But we can at least make sure that we have done our best to produce forest reproductive material with the widest genetic base as possible, and then how this is employed in forest management, I think is another question at the moment. Yeah.
Thank you very much. Any other questions? I don't see any hands up. So thank you very much, Jan Peter, for your presentation. Now we will move on to the discussion part. So I will give the floor to Erik, and Erik, you can handle the discussion from now onwards. Thank you.
So thank you very much, and thank you, Jan Peter. I realize it's far beyond lunchtime in Finland now. So and thank you for everybody else to stay on for the discussion now. If there are any questions or things that you'd like to bring up, please put it in the chat or raise your hand so we can, if there's anything from the people from our audience who have comments or interact. If anybody would like to come into the discussion with a topic to bring in. Otherwise, I would like to, we can start. I would like to start by putting something on the table for you for discussion. And that is, you could say, maybe a bit provocative in the terms of the topic of this theme. So we're speaking about the genetic decline of populations, and I mean, unfortunately, we do see the decline of many species now in Europe. We see it for ash. We heard about ash dieback. We see it for oak. We see it for beech. We see Norway spruce decline. Some of the cases with disease, some of the cases with insects, some of the cases with drought. We see it for silver fir for a long time, European. So my question would be to you: how much of this do you think is actually due to a genetic decline? So how much is driven by a genetic decline, and how much, or is it because there's too little diversity? Or is it because there's too little adaptive potential? Or maybe it's nothing to do with genetics. So this is some reflection about that. So when we see this decline of populations, how do you see the role of genetic decline here? Any reflection on this bit provocative answer question? Okay, nobody will.
Yeah,
No.
I can maybe start. I don't know the answer to that one, Erik. I don't think everything has to do here with the decline, with the genetic. I think the environment is very stressed and stressful, and maybe, I mean, we haven't had enough time to really see the changes. I mean, I mean, if the trees are there despite the stressors, they are plastic enough to be there, and then when they are dying, I don't think we have had enough time to really see if there is potential for adaptation or evolution. I mean, also looking that it changes, and then you know how it is, like 1 year we have drought, the next year we have floods, and then one summer is warm, one is cold. I think maybe it's a little bit confusing for the trees what to do. I don't know.
Yeah, Arne?
I think Darius was ahead of me.
Yeah, but I'll give you the chance first as panel, and then I will.
Okay, thank you. Well, I agree with Marjana. I'm not really sure whether it's a decline for many species, but whether it's a genetic decline,
yeah,
I'm not really sure. It might differ among species there, because for some species like spruce, I'm pretty sure that spruce has been planted in environments where it hasn't been really adapted to the climate change. So it's been planted in the kind of near where it's suited environmentally, and when the climate fluctuates too much and you get these drought periods, it's particularly stressed in these areas, either in the lowland in Central Europe or on pine sites in Northern Europe, for instance. But for the other species, I'm not really sure whether it's just a environmental variation that they haven't really had time to handle yet, or whether it's lack of adaptability. Because when you think of, let's say, Denmark, then I was in Denmark last week, and I heard that for a couple of 100 years ago, only 1.5% of Denmark was covered by forest. Today it's 25%. So there are very many more trees there today than for a long time ago, which must mean that there's a lot of more genetic diversity as food for further evolution and adaptation also in Denmark. And that, I think, regards most countries in the more populated parts of Europe. Thank you.
Okay. And Peter, any reflections?
Yes, I think it's sometimes a little bit difficult to talk about genetic decline as a standalone topic without including the, like, a little bit bigger part, like, for instance, population viability analysis or resilience against future pandemics and future threats, for instance. So the talks that we have heard yesterday, at least I heard the first and the second one yesterday. So all these nice model systems, lizards and rare birds and all these kind of things, where you can even plan genetic rescue at the landscape scale and get nice results and these kind of things. This is all nice, but it rarely applies to trees, I think. This is a little bit of the problem. Nevertheless, that we know from current research that as the area is declining where trees are able to thrive, genetic diversity will decline as well. So there's a very clear mathematical relationship between the area and the amount of genetic diversity. But yet we have absolutely no idea if all these mechanisms that we know, which apply well to really mostly rare plants and animals, also apply to trees, because trees have some delicate features which most of these other species don't have. So they live much longer. They often produce much more offspring compared to a lizard or a bird. for which you have usually a few chicks that hatches from a family and these kind of things. So I think there's really more research needed really on how genetic diversity or genetic decline relates to future resilience. And I think there's still a big gap that we have to cover.
Super. Thank you. Yeah, I think so. My own actually, so one input from myself would also be that I mean, the cases we look at here are cases with very common species with large distribution area, but there are many tree species out there that actually are rare and have smaller population sizes, and also some which are in the phase of having low reproductive success right now, and so there could be something to this, just like something we should consider that. I mean, there are many species, and we are all biased that we focus on the most common species. Okay, yeah, I will open up for. I will list. There will be different talks. There are different people who like now. I have Darius and Francois, and also Mehmeti and Santi, and so. But let's start with Darius and. Please.
Okay, just it's a lot of things to talk about your question, but and issues to rise. But one would be ash, for instance, and like in our project, it would be also nice to involve other countries because this is the species which already had such a huge bottleneck. And from genetic point of view, we could all go and assess what's the diversity left, and it's one good example, for instance, how far it can be shrinked and to do this sort of by genotyping, and then maybe to draw some conclusions, because in some parts that could be also happening with Norway spruce. Because of the bark beetle, and then when you talk about plasticity, I mean there is no plasticity. I mean all are killed and so on. So many issues could be involved in that, and also then to list others. We are here in Lithuania, for instance, getting pressure of all our breeding programs and how we in our breeding programs will cope with the genetic diversity, because material comes from breeding populations where the sample sizes or population sizes are rather limited, like also Arne listed 50 individuals per sort of adaptive environment and so on. So this is sort of another issue that we are discussing, and then basically we are saying, okay, it's basically commercial forestry, it's a rotation forestry. So for in our commercial forest, this diversity should be enough. And then for as a security, we keep our gene conservation forests, as we call dynamic gene conservation units, where we cherish those rare alleles and diversity. So just maybe not so consistent, but some issues from our side. So thank you.
Okay, Francois.
Yes, my question will follow Jan Peter's last point, but also addressed to Marjana. The question is how far the evolutionary potential is really the limiting factor of the evolutionary rate. Maybe the evolutionary rate observed can also be limited by a lack of realization of the evolution rather than a lack of potential. That's one point. So I have two questions to you, Marjana. In the case of maritime pine, actually, you had two populations of the same species, and where I didn't remember if you were able to compare the evolutionary rates in the two and if they were different, if you could have some interpretation of the reason why the evolutionary rate could be, or the strengths of evolution could be different in the two situations. And more generally, there are literature reviews on evolutionary rates in many organisms, but not on trees. That's a pity. And I think the approach you developed is quite innovative and informative on this. Do you have an idea of how many studies of this type do we have today on trees, and if there would be an opportunity already to try to make a kind of review of all these data, specifically on trees, to review evolutionary rates on trees?
Yeah, for the first one, we didn't calculate the evolutionary response, but what we have seen is that even if the trait was under selection, so it had potential because it was heritable, but it wasn't under selection in one or the other environment. So that was, for example, for this P50, which is a very relevant trait concerning adaptation to drought. So the trait was heritable, but there was no selection pressure. So sometimes it's just, you don't need to evolve to another place.
Okay.
And we have seen that that happen. The other question,
Yeah.
okay, the other
Okay.
question.
Already for the first one, this is important because it seems that what is limiting is the selection pressure more than the genetic diversity.
Yeah,
probably.
it could be. It could be that if there is no pressure, then you don't need to evolve. Why? I mean, you save the resources for. Then for the looking at the review, there is not much published on trees. There is a study by Ermin Alexandre on oaks. And then there is the beech study, and then the ones on the one that it soon hopefully will be submitted. But I'm not aware of much more in natural environment. There is a lot of big body of literature on heritability from common gardens and things like that, but not so much in situ, and maybe I'm missing something, but
Okay.
there is not so much.
Okay, I was wondering, for example, in tropical species, which I don't know.
Yeah,
That's why I asked.
I haven't come across any, so yeah.
Okay, is that okay, Francois? Then Santi.
Well, I have a question that is also some kind of reflection, and it comes from the talks in these 2 days, but also my own thinking, and also like a critic to my own work, like when I've been thinking in these topics, and I had a feeling that what we are doing mostly in as a community is to try to document the existence or not, or finding case studies for decline of adaptation. But for me, what is the most relevant question is the threshold that we have to arrive, for instance, for adaptation to take place, like the selective pressure you need for adaptation to. I mean, we have a lot of theories saying that adaptation happens. So, but it's mostly. And this is a bit connected with what Francois said, that what is the selection pressure needed for adaptation to take place. And from another perspective, for instance, when we look to genetic decline, what we see is mostly a demographic decline. And what is the limit of this demographic decline when we should be worried? I mean, can we take, I don't know, 80% mortality, but if there are 20% of healthy trees, that is enough for the population to recover. And for this, there are some theoretical predictions, but I think there is still a lot of work to do to find these thresholds. So I would like to know your opinion, also opinion of the speakers about this, if that would be something that we should put more attention to. And
Any comments? Or you all agree?
I agree very much with what Santi said. So we need to know where these thresholds are, because otherwise we might be worrying for something that we shouldn't. But when something happens, we take it as not important because we just don't know where this. Yeah, I agree.
Super. I will give the word now to my Mette, but just to say that experience with the ash, when we studied ash in Denmark, actually we found, of course, it's a huge decline, and so, but also we found that there's a huge gene flow in terms of seed and pollen. So again, I mean, if you are from a forest perspective in terms of growing ash, of course, it's a huge problem. But I mean, we shouldn't underestimate the evolutionary potential of the species. So I think this is a very relevant question to find kind of limits. Marjana?
Hey, thank you. No, I was just wondering. So one of the topic in this session is also the use of forest reproductive material. So I was just thinking of related to the use of forest reproductive material, for instance, from a seed orchard over a larger area for many years, and if you are using the same seed orchard for many years, let's say 20-30 years, even though you replace some of the clones in the seed orchard, and of course also depending on the number of the clones in the seed orchards. But yesterday we discussed introduction of genetically homogeneous material and how it could influence effective population size. And as you said, Jan Peter, it may be very different in forestry species due to large population sizes, amount of gene flow and long generation times. But is this a topic that we should be looking more into, or are we just very safe with the traits of forestries that they will not be very strongly affected? Just wondering how we should think about the use of forest reproductive material from seed orchards over really large areas, and maybe in vulnerable areas where there's not much natural populations left. Any comments?
Yes, please. Yes, Jan Peter, please.
Yes, I think I have a very clear answer on this. I think during times like these, and also speaking as a conservationist, I think having the highest level of insurance that you can achieve is always the best way to go. So I wouldn't rely on like any things like just transferring material from other regions is one way to do it. That's for sure. But the question is also at one day, I mean, if you have a look at future distribution models of many tree species, you see that the material that we probably intending to move northwards now, so the origins will be probably gone in a few years or in a few decades, depending on the species. So I think putting more effort on really practical conservation is absolutely imperative. So it might be that many of the populations which we see under risk will still persist for decades. There's no question about this. But I mean, why do you do insurance, for instance, fire insurance for your house? Because you rarely ever expect that your house will start burning, right? That's why we do it. So in a very similar way, I would simply go with the highest insurance policy that is possible. If this answers your question.
Yeah, a little bit about the insurance at least, and I totally agree.
Okay. So, Tor?
Yes, thank you, Erik, and thank you to the speakers for very nice presentations. I've been thinking about one thing. I mean, in the north, in Fennoscandia, we have, for the commercial species, primarily clear-cut and planting and breeding system for a few species. And in Southern Europe, exemplified by, for instance, Slovenia, you have mostly natural regeneration. And Arne has explained how we embrace genetic diversity, and I think we should always be open to whether we actually embrace enough genetic diversity, and there is a system for that now. I mean, we can monitor it quite effectively. But of course, there is a level of uncertainty anyhow. So let's say one question is, well, we do know about a lot about the effective population size, actually, as Arne explained. But how can we be sure that we embrace enough genetic diversity in that system? And to Marjana, how do you prepare for the possible genetic decline that comes along with different distribution of species? For instance, I've seen species distribution models for oak in Hungary, and it doesn't seem very good. So is there any intervention planned in case this happens? Thank you. I mean, this is one question to you, Arne. Do we have plans for the possible need for including more genetic diversity or a mechanism? And Marjana, how do you do it in your system when it comes to possible species decline and genetic decline?
Well, do you want me to reply first?
Yep, please.
Well, I'm not a population geneticist, as I said, so this would be from my experience with genetic variants, and what we are concerned about is the additive genetic variants. And that's the genetic variants that is most important for the short-term adaptation. And I've shown these figures showing that a very, very large proportion of the additive genetic variation will be kept from generation to generation, even in small population sizes. So I'm not that concerned about the amount of genetic variation needed for the kind of short-term evolution, which is hundreds of years even. And but I'm more concerned about how we should transfer the breeding populations in terms of what we have done in Norway spruce. Now we have been very concerned about maintaining the kind of regional structure. So we have the next generation breeding populations should be represented by their +3 selected from the same region. But with the rapid climate change now, I don't think maybe that is a feasible procedure to continue. Maybe we need to transfer these breeding populations, and then it would also be natural to kind of transfer genetic diversity or genetic variation between breeding populations as we move them. Yeah, maybe that was a reply for that. But Marjana also brought up this with genetic variants from seed orchards compared to the natural regenerations, and I guess that, or the natural forest, and how that will affect the total genetic variability. I don't really know how, but you know, in the Nordic countries where we have maybe even 50% of the forest area is not managed, it's just naturally regenerating and reproducing. We have billions or so trees not affected by breeding, and when we see that the breeding population do not differentiate that much from the natural population in the first generation, it will probably not be that differentiated in next generation and the third generation either. So I'm not so concerned about that influence either. But that's how far I know it for spruce. I don't know other species with more limited natural populations and more limited genetic variants in the breeding populations. Thank you.
Marjana, are you doing something?
Yeah.
About it?
Well, I mean, Slovenia, we are trying to do things, let's say like that. I mean, most forests, more than 95% is natural regeneration, but we have a lot of disturbances lately, like fires, or we had an ice sleet, and that there, or wind throws, and sometimes natural regeneration then fails. I mean, there is like the crowns get destroyed, so there is no seed, and in that kind of situations we have to plant. And the problem with a country that has been relying really so much on natural regeneration is a little bit that we don't have the capacity to produce the seedlings. We have also lost some of the knowledge, and then, you know, in terms of also what Marimeta was saying, sometimes we have a lot of seed stands. We don't have any seed orchards, but it's often the same seed stands that we are collecting. And when we are facing with trying to put in maybe like a little bit of pre-adapted genes with enrichment plantings, we face legal restrictions, because the areas that match Slovenia climatically in 50 years would be in Bosnia or outside of European Union, and then we have a lot of problems with getting permissions or getting seedlings or seed and things like that. So let's say that we are trying, and for us, with predominantly natural regeneration, we see planting as something that can help us. But there is a lot of obstacles also we are facing by getting the right sources. That the sources that we think are right, and we want to test them and plan them and follow them to see what is happening. But yeah.
Thank you. I mean, we are back to key OptFORESTS questions, really, about availability of seeds and nursery capacity.
Yeah.
I have a question. Thank you very much. Now because I need to keep the time. So Ivan, and then also have Burton, and then also Darius. So please, Ivan.
Yeah, thanks. Thanks for the great talks. This also extends to yesterday's talks, of course. I'd like to go back for a second to more theoretical things, but they have consequences, of course, also in conservation and breeding. So, and mostly my question and comment addresses Marjana's talk, but it has links to the rest. And also echoes Santi's comments. What if we were missing what I would call the conditional neutrality in time? That is, we see here that there are things that are heritable, but not apparently under selection, or there is selection, but no consequences in the distribution of the genotypes in the populations. And actually, properly, one of the things that's happening is that we are used by training to look at things that change over generations, but selection fluctuates within a generation time, even not considering the current climate change. So I don't know, I don't have the answer, of course, but would that be a possibility that we are not looking at things at the right time span?
So people are nodding. We have a challenge there. Any reflections from the panel, or should we take a question from Jonathan?
Yeah, sure. I just kind of have a reflection that I'd like to hear other people's opinions on, tying together different speakers, say, hearing Marjana recommending we should be thinking about making use of multiple mast years to make sure we increase diversity, and what those masts have been subjected to in terms of climate change, or yesterday with Ophélie recommending trying to introduce small amounts of more adaptive traits in our populations without genetic swamping, and then comparing Arne with sort of more Scandinavian areas where we only have a few generations over thousands of years in our tree populations. I wonder if one of the other aspects we should be considering, and making sure we're addressing is the silviculture and timescales as well. So rather than just sources of material, say here in the UK or in Scandinavia, if we're replacing large areas of woodland in one go, and then we wait another 100 years for change. Whereas at the other end of the scale in Slovenia, we have much smaller coops, group selection, and when the forest reproductive material or natural regeneration is made use of, then we're seeing more use of amassed years more frequently. Then does everybody else think we should be recommending operating on a smaller scale within our forests so that we allow this adaptation?
Arne, please.
Yes, I agree with that. We should look at the temporal scale here. It's very important because even in the Nordic countries, we are following this. The silviculture is following this net balance harvest, which means that the forest owner should not harvest more annually than he can sustain the same harvest over hundreds of years. And the possible harvest should increase over time. So that also regards what kind of reproductive material that is used. So we will also always have a temporal variation in what kind of genetic material you use. And that, I think, is very important also for the fragmentation and the distribution of genetic variation in the forests. Thank you.
Okay, so before I have a last question for Lars, but just, and then I think we should probably everybody's getting hungry. I just want to mention, because I put myself on the speaker list, or that with the new EU legislation, there's a lot of focus on using more species. I don't know if you all looked at this, but there's very much focus on using many species and diversifying our forest plantings with many species, which of course, in terms of diversity, is an interesting development. But then again, I think it could open for a discussion here in relation to what we discussed this morning, on that we should make sure when people start to plant all these species that we actually are really focused on really what are people going to plant in all these forests, because then we're actually dealing with minor species, citation minor species which are less frequent. So just to say that this whole idea about thinking about, for some Denmark, there's now seed sources developed for 25 different species, and that's something that we as a society to think about, how we make sure that these are actually supporting a sustainable diversity rather than causing problems. So this is just a comment from my own side, but then I will last comment from Darius, and then I think we should probably stop there. I will, before giving the word to Darius, I'll say there's been some interesting discussion in the chat. So since we don't have much time, just to say that actually in the chat there's also some important points about ongoing research and how to approach the question. But Darius, for last very short question.
Okay, very sorry. I said also we are like in Finland, very hungry, 2 hours post lunch, so we are really. But I just was thinking, as this is EUFORGEN, to raise this issue of management, to coming back out of management of our gene conservation units. Because there are some guidelines, but I think we still need knowledge how to, because this should be like our diversity hotspots. And when then they regenerate, when they have to form this multi-age structure, then there are issues actually for each species, how to do that precisely to encourage and to enhance genetic diversity. And this is the problem which we EUFORGEN could pay more attention in, how it should be done, like gaps then of which size and for each species. So it's a lot of technical issues that we faced with that. And also our gene conservation units overlapped quite often with this nature conservation plots, and then we had a problem with the interventions there. So just a short comment, and yeah.
Thank you. With that, thank you and apologies for going 5 minutes across. But then I'll give the word back to Marjana. I think if you, Marjana, would like to give some final comments.
I think it's Anna Maria. It's
okay.
Yes, thank you very much. So thank you very much, everyone. This was a very vivid discussion. And thanks very much to the speakers that were here today, and also for the speakers from yesterday. I think it was a very fruitful webinar, like with both sessions. So with that, I think that we can close this webinar. Thank you very much. We will see you again, hopefully very, very soon. Have a great lunch and a great weekend eventually. Thank you very much. Bye.
Thank you very
Thank you very much, and thank you for
Thank you.
organizing.
Bye. Thank you very
Bye.
much.
Bye.